order to properly navigate the world, we have to gather some information about it. We have to be able to perceive external stimuli and create some perception about the world around us so we can make appropriate plans. In this lecture, we're going to talk about mechanical sensation. So that would be, of course, touch and hearing. So both of these are going to be based on the opening of mechanically gated ion channels, except for pain. That one's going to be more chemically gated, but we'll lump it together because it still has to do with body sensation. We're going to first distinguish between sensation and perception because they're not always the same thing. And I want to introduce the labeled line principle. This just says that whenever you stimulate the sensory afferents in any sensory modality, it creates that kind of perception. So your retinal ganglion cells will never create anything other than visual perception. Then we'll cover tactile sensation, painful sensation, and auditory sensation. So, based on what's going on out there in the world, that's going to create a different pattern of sensory neuron activity. Those will eventually be relayed from wherever it is they live, whether it's the eye or the body or the inner ear. They'll be relayed eventually to the cortex, in almost all cases going first through the thalamus. There's usually somewhere in the ballpark of about 3 neurons between external sensation and the cortex. And they'll create some pattern of cortical activity, and that would be our perception. Now, perceptions are going to be based off of external stimuli, but the same stimulus can produce 2 different perceptions. Here's a couple of examples shown here. So in the left, you could see a young lady looking away, or you could see an older woman looking to the left. It's the exact same stimuli, but you can create a different perception. And on the right, it could be a vase, or it could be 2 faces looking at each other. So the sensation is going to be our actual detection of the external stimuli. And the sensation is really what we're going to be talking about in this unit. The perception is what goes on in the cortex. That pattern that creates some magical awareness of what it is we're looking at, or hearing, or touching. And that's a little too complicated for a guy like me. So we're going to focus on how we actually sense external stimuli. So the first quantitative study of sensation would be psychophysics. So in this field, they were trying to put numbers to sensation. One of the things that they would look at is the just noticeable difference. Or how different do 2 things have to be for us to tell that they're different? And this is a really important concept, not only in sensation, but also just in basic clinical research. When are things different? When have we made an impact on our subjects? And that's the question we're asking with just noticeable differences. And you can do this for any sensory modality. Probably the easiest and most common test would be the 2-point discrimination test, where 2 points are applied to the skin, and then they're separated by different distances, and we determine at what point are those 2 points perceived as 2 points. And it depends on which area of the body we do this test on. So certain areas are going to be more sensitive to small changes because they have a greater density of innervation. So if we think about the density of innervation of the skin, if we have high density innervation by different nerve fibers, so in this case 5 nerve fibers, and down here let's say only 2. Well, our 2-point discrimination test is going to work if we spread our calipers out, let's call it 2 millimeters or something like that. But if we do that same 2 millimeter test here, let's say we're comparing the lip to the back, we're hitting the same sensory afferent. So we're only stimulating 1 neuron, and therefore it's going to be perceived as 1 point, even though there's 2 of them. We have to spread these calipers out until we actually hit the second nerve fiber, and only then can we perceive it as 2 points. So if you look at the histograms over there, it just shows you the counts of different 2-point discrimination distances. You'll notice the tip of the tongue has a pretty fine 2-point discrimination distance, the upper lip as well. You go to the back of the hand, it's pretty poor. You go to the actual back, it's even worse. And this is just because of different densities of sensory innervation. The just noticeable difference can also be calculated for visual and auditory stimuli pretty well. And it's difficult with taste and smell, but you can blend different percentages of two smells, for example. Clove and coffee or something like that, and ask when do these two smells smell different. It's a little more difficult though, because taste and smell are just so qualitative. It's hard to really quantify those. Now the just noticeable difference can depend on how intense the underlying stimulus is, particularly for vision and sound. Because here we're having to compare two things really using kind of the same nerve fibers, whereas with skin it's all just purely based on distance. But take a look at these images over here. So I created these gratings with the same absolute difference in intensity. All I did was just crank up the brightness in B, so in the bottom. So there's the same absolute difference between all the gratings, but relatively, the relative difference looks very different when you compare top and bottom. And that's because of our ability to distinguish difference depends on how much intensity there is. And this creates that favor fraction. So our ability to detect a difference is going to depend on the underlying intensity. So that change that we're trying to detect depends on how intense it is. Think about picking up a dry erase marker. If I were to make this 1 pound heavier, I would notice. Now imagine that you're going to deadlift, I don't know, 135 pounds. Right? You add a pound to that, you're not going to be able to really tell the difference between 135 and 136 pounds. They'll both be kind of heavy. Add a pound here, very easy to distinguish. So the reason that panel A has such great contrast in it is because the underlying intensity is low. So the same absolute difference appears much larger. So really what the Weber fraction is telling us is that what we're looking at in our CNS is the percent change. This is how we tell difference. Now some sensory modalities are going to be better at detecting small changes like vision. We can detect very subtle changes in light intensity. Sound is a little worse. We need larger differences for us to tell if something is loud, is louder than another. That's probably because of all of the feedback mechanisms that we have. So we have built-in amplifiers in our cochlea, and then we have built-in dampeners from cranial nerves 5 and 7. So we try to keep a relatively constant level of sound coming into our ear there. That might have something to do with it. Now those differences that we see, let's say in different areas of the body in 2-point discrimination or in a visual acuity in the retina, that has to do with convergence of multiple sensory receptors. So let's revisit that 2-point discrimination. That top piece of skin gets more densely innervated compared to that bottom piece of skin. This is going to have much greater acuity because it deals with a very small receptive area. Down here, much larger receptive field. That's what this is, that area of skin that's innervated here, or the visual field that a retinal ganglion cell picks up. So the receptive field is just the part of the world that some sensory afferent relates to. And that world could be the exterior world or your body. We're all a part of the world too. We are the world. We are the children. So the skin here, because we have a larger portion of it innervated, the receptive field is larger. Now, larger could be worse if we're talking about, let's say, 2-point discrimination. So the acuity isn't quite the same. So here we have greater acuity, but down here we actually have greater sensitivity. And that's because we're going to be summing a whole lot more input. We're sampling a larger area of the skin, or if it's the retina, we're sampling a larger area of the retina, therefore a greater number of photoreceptors. So we have a larger dendritic field, and therefore we have a greater number of excitatory synapses. So low levels of stimulus can create enough depolarization to reach threshold. So if we compare that 1-to-1 coupling to a 2-to-1 coupling, you'll notice although we have fewer sensory afferents, they're getting more input from the external world, and therefore they're a little more sensitive. Here's an example. So I'm taking the same image of Arnold Schwarzenegger there. This is from the movie Commando on the set. So on the right, there's enough light, everything's fine. So we don't really need convergence. In that case, we have enough light, we can see the image. So now if we have convergence, all we're really doing is losing our acuity here. The image is a little less clear. Right? So all I'm doing from top to bottom is binning the pixels. And we do this all the time with imaging. If you have very low levels of light, you're going to bin your pixels. And this is why if you take your phone out, which I don't have, and you take a picture of yourself in low light, it looks very blurry. What the phone is going to do is bin together pixels to create a lower resolution image, but to give you enough contrast to still see something. Here we're seeing the loss of resolution on the left. But now let's look at the right. On the right, I dimmed the image. So I reduced the intensity of the stimulus. So we can't really see Arnold Schwarzenegger, but of course we all want to. Now we can solve this problem by binning our pixels. What binning does is combine them together. So we're going to take 4 pixels, and they're going to have different levels of intensity. Let's say it's a, I don't know, something like this. This is going to add up to 40. So fairly low levels of light, if we bin them and put all those pixels together, we just sum their value. Now sure, we've lost our resolution here. We can't tell light in these 4 areas apart, and that's why the picture looks a little blurrier. We're binning our pixels together, so it's going to be pixelated, but we're going to increase the intensity enough that we can actually see what's going on. So in the center of our retina, we have 1-to-1 coupling. That's why we have such great visual acuity there. In the periphery of our retina, we have convergence. And that's why if you want to see a really dim star at night, you don't look directly at it. You look a little to the side so that dim light hits the outer portion of your retina. That convergence then allows us to actually detect that star. Now the different sensory modalities are going to have different types of sensory receptors. Now these receptors could be specialized channels or metabotropic receptors on the axons themselves. So these would be what we call long axon receptors. You'll see these in the somatosensory system, particularly with nociception. You'll see this in olfactory systems. So the olfactory sensory neurons have specialized G protein coupled receptors that detect odorants. You'll see this with taste as well. You'll see it a little bit with vision, but those are very special retinal ganglion cells. They don't actually contribute to our visual perceptions. They help create circadian rhythms and like the pupillary light reflex. As far as the actual visual perceptions, those are going to be from ganglion cells. And ganglion cells are going to be sensory neurons that communicate with some other cell that actually detects the external stimulus. So there will be photoreceptors, then bipolar cells, then retinal ganglion cells. So there's a couple cells between light and the actual sensory neuron that relays back to the central nervous system. You'll also find ganglion cells in the somatosensory system, so non-painful tactile touch. The somatosensory neurons communicate with other cells that actually detect pressure. You'll also find this in our auditory and vestibular systems. So in the inner ear, we're going to have hair cells that detect either movement of the head or vibrations in the air, and they will then stimulate the eighth cranial nerve. So the taste. Taste is actually ganglion cells. I lied to you. They're not long axon. There are taste cells that have the taste receptors, and the seventh and ninth cranial nerves in the tongue are going to be stimulated by those taste cells. Regardless of how these axons are stimulated, when they are stimulated, they create the perception. So whether you're shining light into your eye or rubbing your eye, you're going to perceive something visual. And this is the labeled line principle. So those sensory neurons, no matter what stimulates them, they're going to create that perception because they reliably activate their secondary afferent, and that reliably activates their tertiary afferent, and then usually those go and stimulate the cortex in some way. So the retina is going to project to the lateral geniculate nucleus. The lateral geniculate nucleus is going to project to the striate cortex. And when we have activity in our striate cortex, we see stuff. Our somatosensory neurons, they're going to project, at least for tactile sensation, to the medulla. The medulla will project to the thalamus. The thalamus is going to project to the primary somatosensory cortex. And that's the labeled line principle. We'll see some examples of this. It goes a little bit deeper than that. It actually deals with the specific subset of cells. So for example, if you stimulate a sweet taste cell, no matter how you do it, we're going to perceive sweet. But we'll get to that next lecture. So we'll see some examples of the labeled line principle. Alright, we're going to run through non-painful somatosensation. So tactile perception. This is all about stretch. Whether it's stretch of the skin, stretch of the muscle, stretch of a tendon, it's all about stretch. So here we're going to actually see mechanically gated ion channels. Now, somatosensory neurons, whether they're painful or not, are pseudo-unipolar. So these live in the dorsal root ganglia. They don't actually have dendrites. They have axonal specializations out there in the skin. So here's our spinal cord, one half of it anyway. We've got our dorsal root ganglion here. Now if they're painful, they're going to synapse locally. If they're not painful, they'll head on up the dorsal columns there. And these will head on out into periphery, into peripheral nerves and innervate parts of the skin. In this case, we'll throw it in the fingertip. Bare axons are going to be painful. We can also associate with some specialized sensory receptors that we'll get into for non-painful sensation. But what's going on right here are these generator potentials, where we have some sort of cation channel that detects the stimulus. In this case, it's going to be pressure. So we poke on the skin, that indents the skin. It's going to push on this sensory receptor, and that's going to somehow stimulate this nerve fiber. So we're going to see depolarization of our sensory fiber. If we just look in the dendritic portion of it, so the specialized portion of the axon there, that's associating with our sensory receptor, this is what we'll find. Immediately outside of that sensory receptor, we're now in the axon proper, because here we have our voltage-gated sodium channels. So that depolarization is going to bring us up to threshold, and we're going to fire action potentials as long as we have that pressure. We'll see a change in the frequency depending on the accommodation, but we're going to have some stimulation because of pressure. That's going to open up mechanically gated ion channels. That's going to then stimulate voltage-gated ion channels. to create action potentials. These action potentials then travel back into the spinal cord, either synapsing locally or heading on up to the medulla. So, it's all about mechanically gated ion channels, with one exception. Now, in the periphery, we're going to see a mix of different fiber types. The largest are going to be our group 1 axons, or the A alpha, and these always deal with motor signals, whether it's motor output or motor input. So, for example, spindle afferents, just spindle afferents, and that's it. No need to go beyond there. So the afferents that sense muscle length, that create those fast stretch reflexes, those also use group 1 axons here, because controlling movement is critical for proper nervous system function. So we use the largest and fastest fiber types there. The tactile sensation is going to be A beta for the most part, aside from the muscle spindles. Those A beta fibers are a little bit smaller, a little higher resistance, and therefore a little slower conduction velocity. Those are going to sense tactile sensation of the skin. Then we have our A delta and C fibers that are much smaller. C fibers are also not myelinated, so they are the slowest. Those are both going to create pain. We'll talk about pain in a little bit. Back to just touch. So, the touch that we feel in our skin is created by ganglion cells, and so those ganglion cells have to associate with some specialized receptor. There are 4 of them to choose from, so going from surface to deep, we have our Merkel's discs and Meissner corpuscles. Those are going to be found around the epidermis and upper dermis there, so near the surface. The Merkel's discs actually seem to spit out glutamate. So here we're going to actually be dealing with ligand-gated ion channels, so this is not always true, but everything else is mechanically gated. So it'll be true there. That's all right. Going a little bit deeper, we have our Ruffini corpuscles and our Pacinian corpuscles. So the deeper you are, the more intense the stimulus needs to be in order to actually stretch that area. So very light touch is going to be sensed by Merkel's discs and Meissner corpuscles. The Ruffini corpuscle and Pacinian corpuscles are going to need a little deeper stimulation. So in our shallow and deep sensory receptors, those are going to hook up with different types of afferents. So Merkel's discs, those are going to hit slowly adapting afferents, and the Ruffini corpuscles will hit slowly adapting afferents. The way to remember this is that it doesn't quite make sense. Merkel and Ruffini has quite an R sound, but they're not rapidly adapting. They're slowly adapting, because it would be too easy if the R sound mapped up with the other R sound. The rapid adapting afferents are going to hit the Meissner and Pacinian corpuscles. So when we talk about adaptation, what we mean is a change in firing rate. So this right here, well, that pattern in some cases might look like this, where we see some rapid adaptation, or it might be fairly steady. This change in firing rate, where it's fairly stable here, or decreasing over time, that's the adaptation. Now, rapidly adapting afferents are going to be more responsive to stimulus onset and offset. So you're going to see them more responsive to vibration. Things like movement along the skin. That's going to vibrate the skin, and that's going to be very exciting for Meissner corpuscles, for example. So that subtle movement across the skin is going to hit more surface receptors like Meissner corpuscles and Merkel's discs. But the Meissner corpuscles are going to be more sensitive to that movement because of their rapid adaptation. When the object remains still, those Meissner corpuscles are stimulated only when you first grasp the object, and then they quickly adapt. They are going to be more sensitive to change. And so that's why the movement of an object, even though we're hitting the same areas of skin, is far more stimulating than a stationary object. When it's stationary, we're only getting the slowly adapting afferents. When it's moving, we're hitting slowly and rapidly adapting afferents. And that's because our nervous system needs to be more sensitive to change. Now, if you want to stimulate your Pacinian corpuscles, you're going to need stronger vibrations. So you might need to use some power tools. Or you might need to have a firmer grasp and have it move. Skin stretch, or the sustained stretch of skin, is going to be sensed by the Ruffini corpuscles, and this will help us know kind of limb position based on how stretched our skin is. There will be other contributors to limb position as well, but this is one of them. Now, the way that all of these, aside from our Merkel's discs, work is with stretch receptors. When we push on the skin, we physically deform the skin. That pushes also on the axon, and it actually forces open ion channels. So when we stretch the membrane of the axon, we stretch the ion channels in it. When we stretch them open, their conductance increases, and that can allow for depolarization. That's the mechanically gating. We physically pull open ion channels. You can do the same thing in your eye by applying pressure and rubbing. You'll physically open ion channels. Everything is mechanically gated to some degree. But the stretch receptors that we have in our A-beta afferents are very sensitive to stretch. So they'll wrap their axon into these sensory receptors, like a Pacinian corpuscle, for example, and that will amplify the pressure so that a little stretch gets translated to a larger stretch, and we physically pull open ion channels to depolarize our dendrites. We'll also have afferents hitting hair follicles. Same idea here. It's going to be stretch receptors, but we're going to be using A delta fibers. That's the only real difference. So they're going to surround the hair follicle. When the hair follicle moves, that's going to stretch their axon and pull open mechanically gated ion channels. We'll also sense muscle and tendon tension by using muscle spindles and Golgi tendon organs. Now, when you go by a piece of meat, you're largely looking at extrafusal muscle fibers, but there are very small intrafusal fibers scattered throughout there in sensory organs called muscle spindles. The muscle spindle is going to have sensory fibers that detect stretch. There's a few types of these. There's the type 1A afferents that rapidly adapt, and there's the type 2 afferents that very slowly adapt. But whenever we stretch a muscle, very basically what that's going to do is stimulate spindle afferents. So we can detect roughly the length of our muscles. Let's say here's a muscle, and then we stretch that muscle. Notice it's a little bit longer down here. That stretch is going to be sensed by spindle afferents. Same idea here, it's still mechanically gated ion channels. Rather than sensing skin stretch, we're sensing muscle stretch. So when the muscle fibers are pulled a little bit longer, there are some sensory components in the muscle spindles. That muscle spindle also stretches, and that stretches the axons in it. The mechanically gated ion channels are going to cause the firing of action potentials in our spindle afferents. So we can detect muscle length. This will help create some fast reflexes to cancel out involuntary movements, and it'll also play a role in proprioception. The last sensory receptor we need to talk about is the Golgi tendon organ. Same idea, it's still mechanically gated ion channels, but rather than hitting the skin or the muscle, here we're wrapping our axon in the tendon. So the Golgi tendon organ is going to sense stretch in the tendons. This also helps create some fast reflexes, so if there's a lot of tension on the tendon, that's going to decrease muscle tone so that we don't continue to pull on a tendon and potentially damage it. The Golgi tendon organ is also going to give us some idea of joint angle. So we're going to monitor the length of our muscles. the tension in our tendons, and the stretch of our skin, and those together are going to help us create our sense of proprioception. So a number of muscle fibers are going to feed back into the central nervous system. They'll eventually hit our somatosensory cortex all together, and we'll have some idea of where our body is in space. They'll hit other areas as well, for example the cerebellum. That'll help our cerebellum know body position so we can properly coordinate movement, but we won't be consciously aware of it. Now besides tactile sensation, there's also noxious sensation, pain sensation. Nociception is carried out not by mechanically gated ion channels, but by chemically gated ion channels. And the chemical that we're detecting here is, for the most part, going to be acid. We can also sense temperature too. There are going to be heat and cold sensitive ion channels. So they're going to be stimulated by very hot or very cold temperatures. And then we also have our acid sensing ion channels. So they're going to detect pH. Because when you have tissue damage, there's going to be a change in pH. We'll get to that at the end of this little section here. So there's a collection of trip channels, or transient receptor potential channels. These are cation channels that are stimulated by different things. For our nociceptive afferents, acid and heat. Think about the things that hurt. Acid and heat definitely hurt. Now that acid is going to be caused by edema or swelling. So whenever we have an injury, we're going to create an inflammatory response. That's going to cause some swelling. We'll go over in later how that works. That swelling, though, is going to create a local drop in pH because we have reduced gas exchange. Now, the activity of our trip channels is going to be changed depending on other receptors present on nociceptive afferents. So when we have tissue damage, that's going to be caused by the bursting of cells. So when we have tissue damage, let's say we have a little, I don't know, mechanical injury here, we get a cut on our finger, well, that's going to actually burst open cells right there at the area where we cut, and so their intracellular components leak out. So that boo-boo causes cellular damage. What was inside is going to leak out. So two important molecules here, ATP, bradykinin, are going to leak out. These are going to bind to GQ coupled receptors on nociceptive afferents. That's going to then stimulate, in a very roundabout manner, protein kinase C. Right? Remember, we're going to stimulate phospholipase C. That's going to create diacylglycerol that helps stimulate protein kinase C. It'll also create IP3. That'll, in a roundabout manner, increase intracellular calcium. Anyway, what protein kinase C is going to do is phosphorylate those trip channels and increase their conductance. And it's going to do that by lowering the threshold of activation. So if they're temperature sensitive, lower temperatures will stimulate them. So now you're more sensitive to painful stimulus. So when we have tissue damage, we want that area to hurt. We want that area to hurt so that we don't go touching it and irritating it. This will allow the healing process to be carried out a little more efficiently. Now the fibers that we stimulate are going to be of two types, A delta and C. A delta, although it's small, it's still myelinated, so it's much faster than C fibers because they are smaller and unmyelinated. So that initial sharp pain, that's going to be from A delta fibers. And then there's a longer-lived kind of throbbing pain, also an itchy pain, that's from C fibers. So if you have an area that's damaged, after that initial pain, as the tissue is healing, you'll notice there's an area of redness around it. We'll get into that in just a little bit, but it also itches. So if you ever have a scab forming, and the area around it kind of itches, and that's because of stimulation of C fibers. C fibers create itch. The redness and the edema is caused by the axon reflex. So, let's say we have some damage. We've stimulated our C fibers. How does that lead to inflammation? All right. So, let's get us a C fiber up here. So when the C fiber is activated by pain, it's going to be strongly activated, it's going to be firing a burst of action potentials, and when we hear burst of action potentials, we should be thinking neuropeptides. In this case, we're going to be releasing substance P and calcitonin gene-related peptide, CGRP. These are two neuropeptides that are going to act on a couple of different targets. One of their targets are going to be some immune cells. So the mast cells. These are going to help contribute to inflammation by releasing histamine. Histamine is going to stimulate our C fibers and keep the pain going. So when we have stimulation of C fibers, they create this positive feedback loop that keep them activated. And this is going to help prolong pain. It's also going to make them a little more sensitive, a little more excitable, and so the area is a little more painful. Their substance P, calcitonin gene-related peptide, are also going to act on vascular smooth muscle cells. And what it's going to do is cause them to relax. That's going to lead to vasodilation. The purpose of this is to increase blood flow and allow us to pull out white blood cells because we have an area that's been injured. We also, while we're pulling out white blood cells, we're also going to pull out some fluid. That's what then leads to the edema. So the increase in blood flow and the removal of white blood cells and the fluid that follows causes a bit of swelling. The swelling that occurs here creates a large kind of pool of fluid, and it's kind of like a little cesspool. So the gas exchange isn't as efficient. Because we have this pool of fluid here from this swelling, we have less efficient gas exchange, and so carbon dioxide can build up. That, of course, leads to a drop in pH because of our carbon dioxide buffer, the bicarbonate buffer. That drop in pH is, of course, sensed by, you guessed it, C fibers. And this is going to create the hyperalgesia that we get in an area of damage. Because it all comes down to stimulating those ion channels. Those TRP channels are going to be sensitive to pH. pH drops, they're more active, C fibers are more active. If they're more active, we're going to perceive itch, and when we touch it, it's going to hurt. Don't touch the boo-boo. Let it heal. Let's move on to the ear. So whether it's vestibular sense or sound, it all comes down to, again, mechanically gated ion channels. We're going to be dealing with hair cells here. Now we're just going to talk about sound, so we're only going to hang out in the cochlea. But this is also true in your vestibule and in your semicircular canals. Hair cells are going to sense displacement. That's going to create an electrical signal within them that causes them to then release glutamate. Glutamate is going to stimulate the 8th cranial nerve. So, for sound, we're in the cochlea. Alright, so here's our outer, middle, and inner ear there. So sounds are just vibrations of air. They're going to enter the ear and vibrate the tympanic membrane. That'll get translated through the ossicles there in the middle ear to vibrations of the oval window. That oval window is going to push on the cochlea here. Now let's just unravel the cochlea and have a look at it. So we got our tympanic membrane vibrating on the left. Those 3 ossicles, we're pushing on the oval window, and that moves around this fluid called perilymph. That perilymph is going to vibrate the basilar membrane there, and that's where the magic happens. The round window is there to relieve the pressure, so when we push in the oval window, the round window bulges out, and vice versa. This is a closed system for the most part, so we have a uniform volume of perilymph in there, so if we don't relieve the pressure, it builds up, we might damage our inner ear. We don't want that. Now if you look along different portions of the cochlea, you'll find that it's going to be sensitive to different frequencies of sound. So when you're near the oval window, those fibers of the basilar membrane are much shorter. Here's my oval window. Here's my round window. And here's the basilar membrane. These fibers actually get longer as you move toward the apex of the cochlea. I realize the cochlea looks larger down here at the base, but that's just the bone. The basilar membrane itself, the little strings that are going to vibrate, just like they do on a guitar or a piano, longer strings are going to create longer wavelength vibrations, thus lower frequency sounds. So, at the base, near the oval window, high frequency sounds are going to be most sensitive here because it's shorter strings, shorter basilar membrane fibers, longer fibers at the apex, thus lower frequency sound. And here's the actual recording from different areas of the cochlea. They then record the characteristic frequency of sound that gives them their maximal stimulation. And you'll notice as you move further away from the base, so 100% far away from the base, lowest frequency sound. When you're at the base, down at the bottom of the Y there, the characteristic frequency, much higher. Shorter strings. Shorter strings produce higher frequency sounds. The sensory organ that we're dealing with here is called the organ of Corti. So this is found between the basilar membrane and the tectorial membrane. So let's flip this up a bit. So our basilar membrane is going to be a bunch of strings sitting there. This is going to vibrate up and down as we have sound hitting the ear. We have an inner hair cell layer, and then we have outer hair cells. The inner hair cell is the one that actually detects sounds so that we can hear it. The outer hair cells, there's a row of 3 there. These are going to act as amplifiers. Then we have our tectorial membrane sitting up here. Let me draw that a little bit differently. There we go. So think about what's going to happen here. As our basilar membrane here vibrates up and down, it's going to be pushing hair cells into this tectorial membrane. When we do that, it's going to deflect these little projections, these little stereocilia. It's going to bend them, and that is what creates the electrical energy here. So, those stereocilia are going to be linked by little filaments of protein called tip links. They're made of cadherin proteins. I'm just going to draw 2 here. The reason these are called stereocilia is because they operate in pairs. That's the stereo. The tip link is going to go from one stereocilium on to the next, and it links up with a potassium channel. Now you're probably thinking this would hyperpolarize, and you'd be right if we weren't sitting in endolymph. Endolymph actually has more potassium than the cell. So potassium reverses at somewhere around 0, positive 4, something like that. So potassium is actually depolarizing. It's going to flow in and depolarize the cell. That'll stimulate voltage-gated calcium channels. Calcium flows in. That's going to stimulate release at the ribbon synapse sitting here, so that we spit out glutamate. And that's what will stimulate the 8th cranial nerve. The degree to which we let potassium in depends on how much tension we have on the tip link. If we deflect toward the taller one, the distance increases, thus the tension increases. If we deflect toward the shorter one, the tension on the tip link drops. Potassium influx decreases and we get hyperpolarization. So when we bend our hair cells this way, we depolarize. If we bend our hair cells this way, we hyperpolarize. We're not going to fire an action potential in our hair cell. You only see that in invertebrates. For us, it's just a graded change in membrane potential, and thus a graded change in calcium and a graded change in glutamate. The ribbon synapse here allows us to keep up a high rate of vesicle release. We release about 100 vesicles per second. Luckily, the ribbon synapse has anywhere from about 100 to 1,000 vesicles in the readily releasable pool, and there's a few of these, so we can keep up. So when the basilar membrane vibrates up and down, it pushes the hair cells into the tectorial membrane. That then pushes their stereocilia and creates that depolarization, leading to glutamate release and stimulation of the 8th cranial nerve, the vestibulocochlear nerve. That's going to move through the brainstem, eventually making it to the temporal lobes where we hear stuff. And that's what this slide is telling you all about. So we get pushed up, that moves the stereocilia and hair cells, and they depolarize. Calcium comes in, they spit out glutamate. Those outer hair cells have an additional function, and that is to change their length depending on their membrane potential. When they depolarize, they shorten. And what that does is amplify vibrations of the basilar membrane. So when we push the basilar membrane upward, hair cells shorten, further pulling up the basilar membrane. Notice this relationship here. When they depolarize the outer hair cell, its length decreases. And here you can actually see it in action. So they have a hair cell on the tip of a pipette, and they're just changing membrane potential. And you'll notice that little hair cell there is kind of dancing. It's lengthening and shortening. And those subtle changes help amplify movement of the basilar membrane, and thus create larger potentials here in the inner hair cells. This is accomplished by a protein called prestin. Prestin is embedded in the membrane of our outer hair cells. When the cells are hyperpolarized, the negative charge fills up prestin and causes it to elongate. When the cells are depolarized, that negative charge leaves and prestin shortens. And so that's what causes hyperpolarization to lengthen and depolarization to shorten the outer hair cells. And all that's going to do is amplify that vibration right there, so we can hear quiet sounds. We also have some feedback on the tympanic membrane and the ossicles to dampen loud sounds. So the amplifier lets us hear low amplitude sounds, and those built-in reflexes help protect our ears from very high frequency sounds that might vibrate the basilar membrane too much, leading to excitotoxicity of our hair cells. And once you lose your hair cells, you lose your hearing. And that concludes this lecture. If anything wasn't clear, please fill out the questions box. I'll see you later.